One of the things that mass spectrometry offers us in the high throughput screening space is really the ability to be able to screen very, very quickly in a very, very clean way, so fewer artifacts. And very often we can do this as a direct measurement of substrate to product conversion. And that's great because we don't have to buy extra reagents, we don't have to buy labels and those sorts of things, which obviously add cost. And of course, cost means reduced number of samples that are possible. So in the high throughput screening space, the rapid fire platforms that are now marketed by Agilent are really considered to be kind of state of the art for high throughput screening. Because they're using a simple solid phase extraction methodology rather than an LC, we can process samples much faster, so around about 8 to 10 seconds per sample. But even at that speed, it's really not fast enough. Of course, taking away the LC component to a certain extent also demystifies some of the technology around mass spectrometry and makes it a little bit more user-friendly as well. The aim of the collaboration that AstraZeneca has with LabSite and Waters is really to try and push forwards the boundaries of high-throughput mass spectrometry. Electrospray mass spectrometry is really still, I think, broadly applicable, and although there are some other high-throughput techniques using mouldy instruments, many labs will probably have access to electrospray mass spectrometry platforms. So being able to convert those into a really high throughput technology was really the main aim for this. And the use of acoustics obviously is really exciting because it gives us an opportunity to really use all the advantages of acoustics, so low sample handling and very high throughput potential in a way that's not been really used in terms of coupling it to mass spectrometry in the past. Within AstraZeneca, we've been able to use this technology, even though it's still in its prototype stage, to support our high-throughput screening campaigns. Last year we ran a full collection screen, 2.2 million samples, and to date we've probably processed around about 5 million samples through the platform. So this is something we've never really been able to do in the past with a single MS system. As with any type of research project like this, I think it's probable that we will get significant improvements in sensitivity, which will really add value as we move much more towards a commercial instrument. So I think we're probably still a year to 18 months away from having any kind of commercial offering available, but hopefully we'd like to see it very broadly applied in the biochemical screening space, and we're now starting to broaden out and look at other potential application avenues as well. We'll be sharing some data here around cell-based assays, which will be a first from application with the acoustic mass spec in that area as well. So we're very excited about that, and hopefully we'll see more applications in the future. So PittCon's really important to us because it gives us a really great opportunity to talk to a really broad audience. PittCon's renowned for being really at the forefront of analytical sciences, and really acoustic mass spectrometry is just another analytical tool. So being able to come to a conference such as this one and really be able to talk and access such a wide spectrum of scientific excellence, I think is really important for any project.